The urea cycle. Good day everyone. Welcome back to our channel. My name is Dr. Morabais and I am a professor of biochemistry and molecular biology. I have been teaching biochemistry in medical schools for about 30 years. This channel is created to discuss important biochemistry topics, their metabolic and medical relevance, and especially the high-yield areas that medical students preparing for their exams, including their board exams, need to take note of. Today, we are discussing the urea cycle, a very important metabolic cycle in the body. We shall discuss the urea cycle. 1. What it is. 2. Its metabolic importance. 3. Its regulation. 4. Its associated metabolic disorders. 5. How to manage urea cycle defects. 6. High-yield areas to note. The urea cycle is a series of biochemical reactions that occurs in the body, where it plays a crucial role in removing excess ammonia from the body. Ammonia is toxic to humans. Even minute quantities are toxic to the CNS and may produce tremor, slurred speech, blurred vision, cerebral edema, somnolence, that is, sleepiness during the day, and, in severe cases, death. The body converts the toxic ammonia to the non-toxic urea via the urea cycle. Here is a very important factor contributing to the devastating effect of ammonia in the body. Ammonia combines with alpha-ketoglutarate to form glutamate, thus depleting the cell of alpha-ketoglutarate. Note: alpha-ketoglutarate is an important intermediate of the Krebs cycle. Depletion of alpha-ketoglutarate impairs the Krebs cycle, leading to energy crisis. Brain cells are particularly vulnerable. The urea cycle occurs in the liver. There are 5 reaction steps. The first 2 steps occur in the mitochondria. The last 3 steps occur in the cytosol. Here's a step-by-step breakdown of the urea cycle reactions, highlighting key enzymes, intermediates, and common defects. Step 1. Catalyzed by the carbamoyl phosphate synthetase 1, CPS-I, enzyme. The cycle begins with the synthesis of carbamoyl phosphate from ammonia, as ammonium ion, NH4+, carbon dioxide, as bicarbonate, HCO3, and ATP. This reaction occurs in the mitochondria and is catalyzed by CPS-I. As shown in the screen, note that 2 moles of ATP are consumed. CPS-I is the regulatory enzyme of the urea cycle. N-acetylglutamate is the coenzyme for CPS-I. Step 2. Ornithine transcarbamylase, OTC, is the enzyme. Carbamoyl phosphate reacts with ornithine to form citrulline, which is then transported out of the mitochondria. This reaction is catalyzed by OTC. Carbamoyl phosphate plus ornithine right-pointing arrow citrulline plus phosphate. Step 3. Argininosuccinate synthetase, ASS, is the enzyme. Citrulline reacts with aspartate and ATP to form argininosuccinate. This reaction is catalyzed by argininosuccinate synthetase. Citrulline plus aspartate plus ATP right-pointing arrow argininosuccinate plus AMP plus PPI. Note that ATP is hydrolyzed to AMP and pyrophosphate, PPI. The pyrophosphate is later broken down to 2 inorganic phosphate and releasing more energy to make the reaction irreversible. Thus, the consequence is the loss of 2 phosphates, which is equivalent to losing 2 moles of ATP in regular reactions. Step 4. Argininosuccinate lyase, ASL, also known as argininosuccinase. Argininosuccinate is then converted to arginine and fumarate. This reaction is catalyzed by ASL. Argininosuccinate is broken down to form arginine and fumarate. Remember, fumarate is an intermediate in the Krebs cycle. This step is the link between the urea cycle and the Krebs cycle. Step 5. Arginase, ARG. Finally, arginine is hydrolyzed to form urea and ornithine, which is then recycled back into the cycle. This reaction is catalyzed by arginase. Arginine plus H2O form urea plus ornithine. The fact that arginine is produced in the urea cycle makes it a semi-essential amino acid. It is not essential for healthy adults but is essential for babies, growing children and adults that are recovering from emaciating illnesses because the rate of production of arginine in these people could not match up with their body's demand for it. High-yield areas for medical students. 1. CPS-I deficiency. A common defect in the urea cycle, leading to hyperammonemia and neurotoxicity. 2. OTC deficiency. The most common urea cycle defect, which can cause hyperammonemia, seizures, and coma. It is X-linked recessive, affects mostly male children. There is elevated orotate levels. 4. Classic citrullinemia, a defect in ASS deficiency, resulting in the accumulation of citrulline in hyperammonemia. 3. Argininosuccinic aciduria, a defect in ASL, resulting in the accumulation of argininosuccinate in hyperammonemia. 4. Hyperargininemia, a defect in ARG, causing arginine to accumulate and leading to spastic diplegia and mental retardation. In hyperargininemia, the renal transport protein that transports ornithine, arginine, lysine and cysteine back to blood after glomerular filtration would be saturated with arginine, thus leading to accumulation of ornithine, arginine, lysine and cysteine in the kidneys. Two molecules of cysteine can combine to form cysteine. Cysteine is less soluble and can precipitate to form cysteine calculi, leading to formation of kidney stones. Key intermediates. 1. Carbamoyl phosphate, a key intermediate in the cycle, which is also used in the synthesis of pyrimidines. 2. Citrulline, a non-essential amino acid that plays a crucial role in the urea cycle. 3. Argininosuccinate, an intermediate that is also involved in the synthesis of nitric oxide. Note: argininosuccinate can indirectly generate nitric oxide, NO, through its role in the citrulline NO cycle, which involves the conversion of citrulline to arginine, the substrate for nitric oxide synthase, NOS. Defects in the urea cycle can lead to hyperammonemia, which can cause seizures, coma, and even death. Medical students should be familiar with the clinical presentation, diagnosis, and management of these defects, including the use of alternative paths, such as the ammonia scavengers, to remove excess nitrogen. Note: the glycerophosphate shuttle primarily impacts energy production and cytosolic redox balance. While not directly involved in nitrogen metabolism, its influence on cellular energy levels and precursor availability can indirectly affect the efficiency of amino acid synthesis and other nitrogen-related processes. Likewise, the malate aspartate shuttle, in addition to impacting energy production and cytosolic redox balance, also indirectly affects nitrogen balance by influencing the production of aspartate, which is a substrate for the urea cycle. The urea cycle is the primary pathway for nitrogen excretion in mammals. Management focuses on lowering ammonia levels and reducing protein intake while ensuring adequate nutrition. This includes dietary protein restriction, ammonia scavengers, and in some cases, liver transplantation. Ammonia scavengers like sodium benzoate and phenylacetate, phenylbutyrate offer alternative routes for nitrogen excretion, bypassing the urea cycle. Sodium benzoate reacts with glycine to form hippurate, which is then excreted in the urine. Sodium phenylacetate, phenylbutyrate, these react with glutamine to form phenylacetylglutamine, also excreted in the urine. Other compounds, L-ornithine L-aspartate, LOLA, and ornithine phenylacetate, OPA, are also ammonia scavengers. In summary, understanding the urea cycle reactions, enzymes, and intermediates is crucial for medical students, particularly those studying biochemistry for their board exams. Familiarity with common defects, such as CPS-I and OTC deficiencies, and key intermediates, like carbamoyl phosphate and citrulline, can help students recognize and manage urea cycle disorders. For more information about how ammonia is generated and transported in the body, look out for my video on protein catabolism, deamination, transamination and ammonia transport. Question for you. Why would the deficiency of ornithine transcarbamylase lead to elevated production of orotic acid, orotate, in tissues? Leave your answer in the comment section. Thanks for watching. If you have found this video useful, subscribe to our channel to have access to our other exciting medical biochemistry videos. Until next time, bye for now.